Okay. Share. So it looks like that's good. Okay.
Okay, so just click on that.
Yeah. So if you click on this one. I didn't look at the pictures yet. Oh my gosh, that's from forever ago. Cool. So you'll just click that one, and then yeah, so this one, and it should be recorded. I can't look at them because I was like, it was burning in my desire to watch or to look at the photos, but no, I don't.
So, a friend of mine,
Yeah. Oh my gosh, the duck is here. I didn't even notice. The duck. Who brought the duck? It was you.
Oh.
At least two people from your one.
Yeah. Yeah, yeah.
There used to be.
Oh my God. Well, I told him. So I told him I almost and
Stand right there.
yeah.
Right
Right here?
Yeah, perfect. You got everybody. One more time. Wave.
Wait. I told him I almost put yeah. Sorry. Oh, Crystal's here.
Oh my God, I don't even recognize him.
Wow, what a lovely reunion. Did you know Crystal? Crystal Cooper?
Wait a minute, what?
Yeah.
For sure.
Or Instagram, because she's so young, she doesn't do Facebook. All right, you two, come on now. Get closer to me. There you go. Get the close up.
Oh God, the flash.
And then we get to, there you go.
Now I'm seeing a white light, Jill.
At the end of the tunnel?
Do I follow?
If they thought there's more, you
Oh.
I just turn it off.
Sounds good. I want to see, so I'm gonna stand. I'm not shaking you.
Yeah.
In the year.
So good afternoon to our seminar today. A lot of old friendly faces, students from the past. Our speaker today is Emily Midlowski, who's going to be defending her thesis on rotifers and liverworts. And she's originally from Dearborn, Michigan, and then got her undergrad degree here. And then for about 10 years, she disappeared, but not quite. She was working a lot of jobs. She was a baker for a while, and everybody knows about her baking prowess. And she hiked the entire Pacific Crest Trail alone, which was quite a thing, dodging a few forest fires along the way. And another great adventure she went on was a trip with a few other my former students to the Hudson Bay that was about 600-mile paddle. It was,
Yeah.
but anyway, I promised I wouldn't do any embarrassing stories about her, but so I decided to do embarrassing photos instead. Hopefully, let's see, what's active on? Sorry, need a mouse.
All the mouses. You see it?
So this is young Emily, way back in the undergrad days here, nerdy botanist keying out a sedge there.
Sedgehead. Alex and I were the
sedgehead. How do we get rid of that thing up there?
I'm gonna drag it up.
You might have to get the mouse on.
I'm having trouble getting the mouse to move. Can you get it?
There it goes.
And keying out a dragonfly after the set. And this was on a trip to Isle Royale, and I really like this picture because it really epitomizes her. You know, everyone else, like Alex there, is kind of complaining about the bugs, and here's Emily. Oh, isn't this grand? And then here's a picture from our great adventure, the Hudson Bay. Some former students here. She's pretty tough. And this is 2 months later, which crossed the finish line. Again, there's some old friends here, great group of students went up there. And when we were coming back from that trip, we had to spend about a week in Churchill, where we actually ran a hotel for about a week. The owner was away, and so we got to run it. Emily got arrested for speeding with on the only street in Churchill there. Well, after the Hudson Bay, she was kind of hoping she could work on polar bears, but turned out I said, well, how about water bears and rotifers instead? And so this was her look after I told her that, and then I explained a little bit more about the project. So anyway, she's going to tell you about her research.
Oh, Lord, that's good. That's some quality. Thanks, Dr. Burtis. Oh, man. Good stuff. Well, thank you all for coming. I'm excited to talk to you today. about my research, looking at rotifers and Frulania. And thank you again, Dr. Robertus, for that introduction. But I also just want to let you know that if you put honey out in the woods, you will attract a Robertus. It was good fun. That trip was great. Okay, so let's see. I'm just gonna turn the lights off because we have some cool images that we're gonna look at throughout the presentation. So maybe they'll dim. But just as a presentation overview, first in chapter 1, I will talk about a synthesis of data that was collected by students in the Robertus lab a number of years ago and describes rotifer occupancy and also some seasonal variation within the lobules. And then the second chapter is more of an experimental study where I tested the hypothesis that rotifers provide Frulania with nutrients. So rotifers can occur across terrestrial and lacustrine habitats in brief temporal windows where they occupy standing water in ponds, pitcher plants, tree holes, and water pockets of soil and moss. There's well-documented research about rotifers in freshwater ecosystems, and in these habitats, the populations can be influenced by bottom-up and top-down processes, depending on the quality and quantity of food available, the abundance of predators, and also water chemistry. But aside from their ability to utilize variable food resources, rotifer population success can be attributed to their life history strategies, which allow them to meet fluctuating habitat conditions. And during these conditions, rotifers can suspend non-essential functions for extended periods of time, which may result in population stability in some terrestrial environments. Deltoid rotifers in particular are adapted to living in variably wet habitats by entering cryptobiosis, which is a state that allows them to survive periods of desiccation. Cryptobiosis is accomplished by an initial contraction where the cephalic and caudal extremities are withdrawn into the trunk and the tissues and cells become compact. This body state is called a ton, and it looks like this photo here. So this is showing a fully hydrated and extended rotifer, and then as it goes into this ton state, it becomes compact into this little structure here. After the drought period has ended and the bryophyte becomes wet again, the rotifer absorbs water and normal activity resumes. So my research today will discuss the rotifers that are found within lobules of an epiphytic liverwort called Frulania eboricensis. I'll refer to it throughout the talk as just Frulania. And here in this little photo, you can see the lobule of a Frulania sprig and a little pink ball. That is the ton of a rotifer. Okay, so Frulania is a pioneer on a variety of trees in the UP, but can be found growing on hardwoods like aspen, red oak, paper birch, and maple. Frulania has those bowl-shaped ventral lobules situated there for water storage during drought periods, and also provide a habitat for rotifers. So in 2004, Putterbaugh and others sought to determine the distribution of rotifers occupying lobules and concluded that there was a greater proportion of rotifers occupying lobules on the exterior of the plant rather than the interior lobules. My study provides a more detailed description of rotifer occupancy within lobules, but before jumping into project objectives, I just want to show you an example of what this system looks like under the microscope. And so in this video, you'll see a typical scene that I encountered throughout pretty much this entire project. So here is the lobules that I've been talking about, and then here are 4 rotifers that you can see kind of beating their little cilia and generating some currents and bringing in some food to consume there. So really tiny things that we're focusing on today. And so some objectives of this chapter were to first examine the density of rotifers found within lobules growing on different tree species in the UP. The second was to investigate the distribution of rotifer occupancy within Frullania lobules. And third was to determine how rotifer populations fluctuate throughout the seasons. And so to do this, throughout the fall of 2014 through 2016, and then again in 2018, Frullania was collected at multiple collection sites around Sugarloaf. The sampling area is located near the south shore of Lake Superior here in Marquette, where the dominant soils are classified as well-drained spodosols. And Lake Superior influences the climate, which typically results in cold and snowy winters and warm summers. Monthly temperatures typically range from a max of 24.6 degrees C in July to a minimum of -5.4 degrees C in January. And so during this time, Frullania was collected from both sugar and red maples, white birch, northern red oak, and trembling and big-tooth aspen. Frullania colonies with green tips on the exterior of the plant were selected. So looking here, this is a typical colony of Frullania. So the green tips that you see were those targeted for this sampling period. I'll refer to these green tips as sprigs. And these sprigs were 5 to 7 millimeters in length with or without branching. And the length was selected because it usually meant that there were 20 to 30 lobules on the sprig. And all of the branching means is that along this primary axis, there is a branch here and a branch here. That'll come into play in a moment. So after sprigs were collected, rotifers and lobules were counted. And we also noted rotifer social groups. So were rotifers by themselves in lobules? Were they present in pairs? Were there 3 or more? And then for the second objective, to determine the distribution of rotifer occupancy within lobules, the sprigs were a little bit longer and had 1 or more secondary branching present. The sprigs typically have clusters of developing lobules near the tip. So it's most common to find really tiny developing lobules here and less so further down the stem. And so each lobule was assigned a sequence number based on its location from that apical meristem. But this protocol designed the first lobule as the minimum size that a rotifer, or that could accommodate a rotifer. Sometimes the main axis had up to 49 lobules and secondary branches had up to 20. Rotifers and lobules were counted and then later converted to a presence or absence for analysis. So in 2015, sprigs were collected on a mostly bi-weekly schedule, but only a few winter samples were collected. So in 2018, sampling efforts were focused on filling in those gaps missed in 2015, particularly in the spring and winter. Sprigs were collected from aspen trees because more rotifers were observed on aspen, and by doing this, we could eliminate one source of variation. 10 to 20 trees were opportunistically selected per sampling date, and 1 to 2 sprigs per tree were collected. There was caution to avoid resampling the same trees and only sampled intact Frullania that had not been previously sampled. And so back in the lab, sprigs were staged on moist paper towel, transferred to a microscope slide with a drop of water, and examined under a microscope for rotifers. For this analysis, rotifers were counted within a 20 to 30 minute period per sprig, but during the winter, because a lot of rotifers were in their ton stage, rotifers were given 5 to 10 minutes to acclimate and hydrate, and tons that failed to become active were not counted. The total number of lobules were counted, and rotifer counts were converted into a count per lobule. Okay, so the results. So using a linear mixed model to model the mean number of rotifers per lobule along those 5 different tree species, we used a date of collection as a random effect to control for different sampling periods. and constructed a frequency histogram to show the number of rotifers per lobule for each tree species. So looking at those results, you can see that the highest density of rotifers in lobules occurred on aspen and red oak. Mean rotifer densities on aspen were significantly different than all other species except red oak. And there's a couple ideas for why this may have been the case. So rotifers' minor preference for aspen is less striking given the modest difference in rotifer densities across all tree species, even on paper birch, which is considered to be a poor host for epiphytes. Differences in density could be due to bark characteristics, but the relationship is difficult to attribute directly. Frullania grows in dense patches on aspen, but there's not much consistency across tree species. Liverworts growing on bark are affected by a number of factors, such as bark moisture, pH, surface topography, microbial communities, the climate of the surrounding forest, all of these different things. And Frullania is typically found on bark with a pH of 5 to 6.8. The average pH of aspen bark is 4, but can be as low as 2.3, and has been reported as high as 6.5, compared to sugar maple, which has an average of 5.5. But these things don't necessarily reflect the stem flow pH that rotifers and their food sources are exposed to. But there's not much research investigating how rotifers in terrestrial environments are affected by these sort of environmental factors. But rotifer communities in peatlands are fairly well studied and show that they are influenced by environmental conditions like pH, conductivity, and moisture. Future studies could examine and measure stem flow pH of different tree species and assess whether chemical characteristics of stem flow influences rotifer densities and also Frullania growth. So using that same data set as the previous analysis, the social groups of rotifers within individual lobules were quantified. So here we found that 61% of lobules had one rotifer present. The frequency of lobules with larger groups just declined exponentially. About 5% of occupied lobules had 5 or more rotifers, with a maximum of 11. It's totally wild to think about. These lobules are so tiny, and that is just so many individuals kind of crammed in one little cup. But at the population level, 34% of the population was solitary, 23% were found in pairs, and 18% in trios. Only 17% of the population occurred with 5 or more. So why be in these groups? Are there any advantages? So considering that about 2/3 of the rotifer population consisted of clusters of 2 or more individuals within a lobule, the previous study that I had mentioned by Putterbaugh speculate that groupings could be attributed to parthenogenic reproduction within lobules or perhaps habitat preference. So rotifers could be utilizing lobules as a means for parthenogenic reproduction, as they are suitable habitats for when conditions are favorable. The lobule has water, it serves as protection from predators. Offspring are able to feed together, kind of generating a stronger current and maybe bringing in more food. But another advantage could be that by congregating together, rotifers may be better able to survive periods of desiccation. There was a study by Hess, Spells et al. That showed that the ability to survive desiccation was strongly correlated with rotifers aggregating together as a way to reduce evaporation. However, with all this said, the available literature on rotifer social behavior is pretty scarce, but this is a great system for experimentation because rotifers can be manipulated fairly easily with some practice. So looking at rotifer occupancy, A binomial generalized linear mixed model was used to model the probability of rotifer occupancy, or excuse me, the probability of occupancy by rotifers as a function of lobule sequence from the apical meristem of the Frullania sprig. And this was done for the primary and the secondary branches. The lobule position was recorded as a sequence number from the apex. So if we look at the results of that, we can see that the probability of occupancy was significantly related to sequence and branching level. The interaction between sequence and branching level was not significant. Rotifers have a higher probability of being in the lobules near the apex, particularly in the first 15, and steadily declined further down the sprig. Secondary branches have a lower probability of occupancy. And so using this image just as a reference, so basically rotifers were found in lobules closer to the tip and less so in these branches out here. So why the preference for younger lobules? One hypothesis for why rotifer occupancy in lobules is highest towards the tips of these sprigs is to avoid predators. So the center of Frullania tends to be a bit more densely vegetated, hosting a greater concentration of tardigrades, nematodes, orobotid mites, all potential predators of rotifers. But also the tendency of rotifers to occupy young lobules suggests that the lobules serve a complex purpose beyond just a habitat. The youngest lobules and leaves, or excuse me, the youngest tips appear to display the highest levels of photosynthetic activity. Following drying events, bryophytes experienced notable nutrient loss due to leaching. Perhaps rotifers are benefiting from this well-oxygenated environment in the lobule, or maybe they feed on microbes breaking down any of the leachates in that environment. In chapter 2, I discussed the likelihood of rotifers providing Frullania with nutrients, specifically nitrogen, from their feces, but a mutualistic relationship was neither confirmed nor excluded. Random colonization could also be attributed to these patterns, but perhaps rotifers are distributing or dispersing to avoid bacterial or fungal parasites. After a period of desiccation, while they're in their ton, rotifers can be dispersed by wind and colonize a habitat in the absence of any sort of pathogen. And in older lobules, fungal hyphae and cyanobacteria were occasionally detected, potentially rendering the lobules toxic or uninhabitable by rotifers. Experimentation or spatial modeling may reveal definite patterns specific to rotifers living in lobules, and also utilizing carbon-13 isotope analysis could show whether carbon from Frullania is obtained by rotifers. So looking at rotifer populations throughout the seasons, to do this, the number of rotifers per sprig was converted to density per lobule on the sprig, and trends in the population were fit to a cyclical GAM regression using the MGCV package in R. The cyclical fit accommodates a wrap between years, so the beginning and the end of the trend line match. So looking at the figure here, we can see that overall the population was relatively stable across the 1.5-year period, but the counts were super variable at the sprig level. So all of the red and green dots on that figure are showing individual counts along the sprig with quite a bit of variation. The coefficient of variation for the mean sprig count was 90.48%, and counts hovered at 0.5 to 1 rotifers per lobule on aspen year-round. If we put the same information on a centered scale, we can see that this scale highlights the deviations from the long-term mean and shows a rise in the late fall into early winter. So in October, you see a spike into November, December, and again throughout January. Okay, so looking at for why this may be. So these results indicate that rotifer populations living in these lobules are fairly stable throughout the seasons in these tiny lobule habitats, allowing limited room for competition or predators. The mean number of rotifers per lobule increased in the late autumn to early winter and remained constant throughout the spring and summer. And rotifers living in the soil show a similar trend. In the soil, they tend to increase in the fall around September and then remain pretty constant through mid-May. The autumnal peak could also be due to an extended lifespan, which has been associated with cooler temperatures and restricted food availability. Bryophyte-dwelling rotifers seem to experience less population fluctuations than those in aquatic environments. In temperate and subtropical aquatic environments, the majority of rotifer populations experience bottom-up control, where populations increase with summer warming and food availability, but then fluctuate due to top-down controls from competition and predators. The abundance of rotifers in the winter was pretty surprising, but it is crucial to note that these were capable of being revived when re-wetted, often assuming normal behaviors within 20 to 30 minutes. So the ability of delloid rotifers to enter their ton allows them to survive unfavorable abiotic conditions, such as low temperature and the absence of water. The average January and February low in Marquette can be between -11 and -14 degrees C, depending on the proximity to the lake. Unpublished data from the Robertus lab shows that rotifers can withstand temperatures up to 196 degrees Celsius, which is the temperature of liquid nitrogen, for 30 days with no significant mortality. And also, pretty wildly, rotifers in Siberian permafrost have been revived after being re-wetted 24,000 years after being in a cryptobiotic state. So the ability of rotifers to undergo cryptobiosis can stabilize the rotifer population year-round during these periods of drought or extreme freezing events. And so to recap the results of chapter 1, these results lay the groundwork for further investigation of environmental effects on the ecology of rotifers and Frullania. Here, the results show that rotifers are common within lobules across tree species, though aspen shows the greatest densities. Rotifers have a preference for younger lobules closer to the apical meristem of the plant. So in chapter 2, I aim to illustrate the potential for rotifers to provide Frullania with nutrients, suggesting a mutualistic relationship. And so if we look a little bit about previous studies that document these tiny animals living in association with them, they've shown that other metazoans may be a significant source of limiting nutrients like nitrogen and phosphorus for the surrounding environment, and also in pitcher plants. Tangentially, Hess et al. In 2005 proposed the zoophagy hypothesis in another epiphytic liverwort, Pleurosia purpurea, seen here on this arc. Hess found evidence that protozoans and other metazoans like tardigrades and rotifers, some nematodes, became trapped in the water sacs of Pleurosia by means of a trap-like structure on the water sac. So in their paper, they showed this image of the water sacs, and then these little black shapes are the ciliate trapped inside. The authors suggest that this hepatic may be acting like a mini carnivorous plant, where they benefit from the work of bacteria within those water sacs, but they didn't document any nutrient transfer to the plant, and they at a molecular level, nor did they show that metazoans are attracted to the plant for any particular reason, other than maybe to feed on bacteria that are associated with it. And this can kind of be related to Frullania in a way that when Frullania is fully hydrated, those lobules are wide open, rotifers can come and go as they need, but when the lobule dries up, it tends to flatten and fold inward. That could potentially trap the rotifers. So dead rotifers or rotifer fecal matter is a hypothesized nitrogen source that could also be absorbed by Frullania. But there has been no evidence of nutrient transfer published. But given the small scale of both the rotifers and the plant, I needed a way to track and observe rotifer-derived materials into the plant. And so to do this, I used quantum dots. And so a little bit about quantum dots. So quantum dots are these man-made nanoparticles with a nanocrystal core, typically of a heavy metal like cadmium and selenium, with an outer coating to stabilize the core and improve the optical and physical properties of the material. The core of the quantum dot can range anywhere from 2 to 6 nanometers, where the size of the core determines the wavelength of light that's emitted and that you use to excite the core. The uptake of quantum dots can be observed using differential interference contrast imaging, or DIC, with a confocal microscope. Other studies have shown the transfer of quantum dots to higher trophic levels, from bacteria to primary consumer microorganisms, but also in vascular plants, where quantum dots were taken up from the soil and transferred to herbivores. Kim et al. Showed that quantum dot uptake from E. coli to a nematode, where they were able to visualize the quantum dots very well in the nematode gut. So this was their control that shows fluorescence, but then in their quantum dot treatment, you can see that the E. coli that were ingested fluoresced really well in the nematode. No studies have shown the quantum dot transfer from an animal back to a plant. So the development of a methodology to understand whether leafy liverworts can take up quantum dots from other organisms can help to understand the complex relationships that liverworts have with their microorganismal inhabitants. Okay, so my objectives for this next phase was to first develop a method for determining whether quantum dots can be transferred across trophic levels, and then 2, use the method to show rotifer-derived feces transfer to Frullania. So in chapter 1, we determined that rotifers have a preference for the green tips of Frullania growing on aspen trees. So I sampled Frullania from aspen throughout September through December of last year. I removed bark chunks as opposed to just the plant because I wanted to ensure that the plant would remain photosynthetically active throughout the duration of my experiment. And I also assumed that rotifers would be present in the samples that I collected. I organized my samples into 2 groups: samples exposed to unlabeled E. coli and samples exposed to quantum dot labeled E. coli. I kept these samples in the greenhouse here at NMU and set them up under an automatic mister to water them and let that go for a week. So the study by Kim et al. Showed great success with the effectiveness of different concentrations of quantum dots in organisms. And so based on their results, I used a 15 nanomolar concentration of quantum dots in my experiment and followed their labeling and suspension methods. So to inoculate the E. coli with quantum dots, I put them both in a test tube and put that test tube on a shaker for 12 hours in a warm room. And then I plated the solution on an agar and LB mixed plate and let that incubate in the dark for 24 hours. And then the unlabeled E. coli were plated on just nutrient agar. So after the bacteria were grown up on this plate, I needed to make some rotifer food. And so to do cell suspensions, I swabbed both plates for bacteria and then suspended them and washed in a solution of phosphate buffered saline, and then suspended the final pellet in spring water. This was my rotifer food. To feed the rotifers, I distributed 100 microliters of both the unlabeled E. coli and the quantum dot labeled E. coli to each bark sample once a day for 7 days. After the 7-day feeding period, all bark samples were removed from the greenhouse. And then in the lab, I removed 1 sprig from the bark sample and examined it for active rotifers using a dissecting microscope. Sprigs containing rotifers were placed into one of those 8 well-chambered well slides with spring water. And following that, we did some microscopy. So Frullania sprigs and rotifers exposed to unlabeled E. coli and Frullania sprigs and rotifers exposed to quantum dot labeled E. coli were both imaged using DIC imaging with the same wavelength of light that would be used to emit fluorescence from the quantum dot core. And so when we imaged the samples exposed to the unlabeled E. coli, we found that there was no fluorescence, which is good. That's what we would expect. But also just noting that no other plant structures or structures in the rotifer were fluorescing under this excitation wavelength. So here's a rotifer within the lobules, no fluorescence. That's great. So then when we imaged the quantum dot exposed samples, we found some fluorescence, which is awesome. It's super exciting. So here's a rotifer showing a little bit of fluorescence in the gut. Here's another rotifer showing a little bit of fluorescence in the gut. And then of course, the plants also showing a bit here in the stem parenchyma and in this underneath. So this result was exciting and indicated that the rotifers can consume quantum dot labeled E. coli, and it also shows up in the plant. But just to be sure that rotifers do indeed consume quantum dot labeled E. coli, Dr. Autumn and I spent some time imaging some live rotifers, which was quite the task. But we did manage to get some pretty wonderful images, quite possibly the most remarkable being this one. So in this particular well, I pipetted 20 microliters of quantum dot labeled bacteria just to give them a buffet. And this particular rotifer gorged. So the concentration of fluorescence in the stomach and also in the intestine just indicate that the rotifers do actively feed on these things when given the opportunity. When I isolated a rotifer from the quantum dot exposed samples, you can still see some fluorescence here in the gut as well. Okay, so then when we imaged the plant, we saw some pretty cool things as well. Let's see, so we found that the stem parenchyma, particularly near the lobules, had very strong fluorescence. We also observed fluorescence in the cell walls of leaves. Looking specifically at the oil body, of the lobule, we can see that despite the fluorescence within the cell walls, there's also fluorescence in the oil bodies inside the cells of the liverwort there. And also in these interesting channels along leaf margins, we saw some fluorescence. So these findings were pretty exciting. They allowed us, they showed the potential for rotifers to provide the plant with quantum dots from their feces. But then the more that I talked with my committee members about it and some other folks, we kind of decided, well, maybe bacteria are attached to the outside of the plant. I also learned later that endophytic bacteria can get into oil bodies of liverworts inside the cells. So I was like, okay, we need to eliminate the possibility that bacteria are influencing this. So I conducted another experiment to reduce that likelihood, or at least try to. So I repeated the methods of labeling E. coli and feeding rotifers, but this time after the feeding period, I removed the quantum dot labeled rotifers from their samples, rinsed them in deionized water to try to get rid of any bacteria that may be stuck to them or to my little rotifer wrangling device, and then put them into a well slide with fresh Frullania and let them sit for 24 hours together. So I think I ended up putting 10 rotifers in each well with 1 sprig of fresh Frullania. And when we imaged these samples, we saw some pretty strong fluorescence. So this image is an example of a sprig that we saw that had just been exposed to rotifer, quantum dot exposed rotifers. And so we see strong fluorescence in the lobules and the stem parenchyma, not too much in the leaves, but very, very much a good signal. And so, like, while this was really exciting, I learned a lot of other things about how bryophytes respond to heavy metals, but more on that in a moment. So overall, the internalization of bacteria-derived quantum dots was evident in rotifers, a finding that was good because it was consistent with other published studies examining the uptake of quantum dots from bacteria to consumer and the assimilation of quantum dots to higher trophic levels. But noteworthy here is the uptake of the quantum dots by the Freelonia. So this is the first study to document transfer of quantum dots from a bacteria to an animal back to a plant. So the results show that quantum dots are absorbed across the entire plant surface, with the strongest quantum dot fluorescence observed in cell walls of lobule parenchyma, in oil bodies, stem parenchyma, around the lobules, but also in the cell walls of leaves. But why is fluorescence observed all over the plant and not just in these like concentrated areas where rotifers tend to hang out? So unlike vascular plants, leafy liverworts lack a cuticular barrier and absorb water and nutrients via osmosis from external capillary spaces, the underleaves, and also in the lobules. The large surface area for water and nutrient absorption is facilitated by a cation exchange capacity in the cell walls. This cation exchange capacity has an important role in the uptake and sequestering of molecules, but only select particles can be passed through the cell wall. Previous studies have shown that bryophytes have an affinity for heavy metals. Particularly Brown and Bates explain the capacity for a metal to bind to a cation exchange site is dependent upon the number of available sites. This can be influenced by the concentration of the surrounding solution and the concentration inside the plant, the elements that are already bound to these sites, and the binding site itself. If the concentration of the water containing quantum dot labeled rotifers excrement surrounding Freelonia was higher than the concentration inside the plant, it's possible that the osmotic balance was enough to be tipped and cause the cadmium to just flood these channels. Intracellular cadmium uptake can displace cations in solution, resulting in an affinity for cadmium. Freelonia also showed evidence of rotifer-derived quantum dot uptake from Thesis, making it possible that rotifers could provide Freelonia with nitrogen through their feces. Nitrogen is the most limiting macronutrient for a lot of plants, despite its essential role in various biological processes like the synthesis of proteins, DNA, RNA, photosynthesis. But nitrogen can be absorbed from nitrate, nitrite, ammonium, and also in urea. Liverworts and other epiphytic plants like bromeliads have been shown to derive nitrogen from organisms in their environment and in the microorganisms that inhabit the water pockets between the leaves. So if we think back to the results from chapter 1, there was a high proportion of lobules that were occupied. It was about 1/3 of the lobules sampled were occupied by rotifers, and the density of rotifers per lobule can be fairly high. So the likelihood that rotifers provide the plant with nitrogen is fairly feasible. However, rotifer seasonality should be considered when investigating the capacity Freelonia has to utilize rotifer-derived nitrogen. Lobule dwelling rotifer populations are active in winter and show an increase in the number of individuals. There are a number of different avenues for research that could expand on this to further understand the nuances of the complexities that are going on here. And just to name a couple, researchers could use infrared spectroscopy on quantum dots before and after digestion by rotifers to understand how the structural integrity of the quantum dots changes post-digestion. Using another form of nanotechnology could be another possibility. Carbon nanotubes could provide another traceable mechanism to identify rotifer-derived materials within Frullania. This might be a more realistic approach as opposed to the heavy metal-based quantum dot. So in conclusion, throughout the duration of this experiment, the constant micromanipulation of rotifers shows that they're able to withstand disturbance and survive digesting cadmium-containing quantum dots that were otherwise thought to have severely toxic effects to many organisms. This research is the first step towards establishing a method in which rotifer-derived materials utilized by the host plant can be tracked. The findings in this study support the idea that Frullania can absorb and make use of nutrients and rotifer feces, and also paves the way for many to discover the complexities that exist between rotifers and Frullania. So in closing, I just want to leave you with a quote from a book that was written by Robin Wall Kimmerer. It's called Gathering Moss. In the preface of her book, she writes about seeing the world through moss-colored glasses. So she says, learning to see mosses mingles with my first memory of a snowflake. Just as the limits of ordinary perception lies another level in the hierarchy of beauty, of leaves as tiny and perfectly ordered as a snowflake, of unseen lives complex and beautiful. All it takes is attention and knowing how to look. I found moss to be a vehicle for intimacy with the landscape, like a secret knowledge of the forest. So hopefully after this talk, you can take this and look at the world a little bit differently and maybe investigate some of those tiny little things that we just blow by when we're out hiking. And with that, I have a profound appreciation to so many people. This project has very much been interdisciplinary and totally outside of my realm. So I appreciate all of the conversations and help throughout the way. And yeah, with that, I open the floor to questions. Yeah.
Throughout the talk, you referred to your experimental animals at phylum level.
Yeah.
I was wondering what species of rotifers, or if we have more than one species, and what the taxonomic identity of those rotifers might tell you about their abundance patterns.
Totally, I'm so glad you asked. So what I didn't talk about were the DNA extractions that I did, which are sort of telling. So I did some DNA extractions that, when put against the rotifer database that is available, showed that the species in this system is Abrotrocha, which is actually in Philodina. So originally I had come into this project thinking that these rotifers were in Abrotrocha, a totally different family. But when we put the sequences that I got up against the sequences available, the similarity was about 90%, which wasn't great. I think I would feel more confident giving it a name if it was like, 98, 99% similar. But I think part of that is attributed to the lack of available sequences in the
Species?
Yeah, so two came up. So one was Abrotrochlespa, and the other one was oh gosh, what was it? It came up with one other one, and I'm blanking on the species now. But I thought it was interesting though, because like I thought they were supposed to be parthenogenic, and so to get a couple, like, oh, they came off the same sprig. Why am I getting two? So curious. Question?
So
Yeah.
you mentioned their reproductive characteristics.
Mhm.
And so when you find more than one in one place, could they possibly just be clones?
Yeah, I mean, possibly. So that's why I was a little surprised to see the genetics come up with different like results. But with that being said, when I sampled the rotifers for the extraction, I didn't pull from a lobule, like I pulled from different sections of the plant. But I think that is another like question that is totally worth asking. So,
You said that when you put the people line and you took it, you said that some of it was with the quantum dots and some of it wasn't. But you put them, when you put them, you did the quantum dot labeled E. coli on agar and Olivia labeled, but then the other ones that were unlabeled, you know, just make sure.
yeah, so the protocol that I followed for the quantum dot labeling, specified that they grew their quantum dot E. coli on a specific blend of LD and agar, so I just followed what they did, because it worked for them. And then the E. coli that I use just to grow on regular nutrient agars, and stick with that. And then
Yeah, this may be a dumb question, but I'm just kind of curious how they move, how the rover moves from one wheel to another. Are they all, my understanding is that they kind of cilia swim around, but can they evolve?
yeah, so primarily under the microscope, when I'm looking at these guys, they're moving around, but sort of crawling and inching. So they have a foot that will attach to a substrate, and then they kind of just like inch like a little inchworm around the bryophyte. But I've also seen them under the microscope, like using that ciliated corona to like do like circles. They'll attach with their foot and then they just kind of like do spins. But they're not necessarily like moving around in that capacity. But then also when they're in their tons, if they for some reason are like, you know, on a leaf of a Rulania plant, they can be washed by passive transport, gravity, any sort of like strippings. Yeah.
So you mentioned like you saw some distinct differences in seasonality in particular. Do you think that's more like environmental driven? And if so, would that mean like some of the microtopography of where these liverworts actually are that have a strong impact as well?
Yeah, that's a really good question. Well,
I meant to ask you a really easy one.
Yeah, to be honest, I don't know. I'll be real. I think that the system that this grows on is so complex and there's so many moving pieces that it's hard to isolate one thing attributing to this thing that we're trying to get at. I mean, with that said, like, you know, I think that microbial communities play a huge role in a lot of this. And like, I think the stem flow chemistry will also play a role. But I mean, even finer scale, like the chemistry of what is going on in that lobule is so curious to me. Like, who's in there, like smaller than a rotifer? And, you know, is the plant giving any sort of leachate? I don't know if that answers your question. Okay.
So I have a point of clarification. So you found more rotifers on the populace sampled Rulania. You mentioned that Rulania occurs more densely on populace. Does that mean more poplar trees have Rulania, or on an individual poplar at Sugarloaf?
Yeah.
There's more for Rulania occupants.
I'm glad that you asked that question. So two parts. So the dense part that I was talking about is that on aspen trees, Rulania colonies are pretty much like just covering the bark. Like they are very abundant on one tree. But also when you go into an aspen stand, you'll see that like these colonies are present on all of the, like almost all of the trees in the stand, as opposed to a stand of mixed hardwoods. You might find, you know, a couple patches on a maple, but then a bunch on an aspen, and then you go over to a red oak, and you're like, oh, there's kind of a lot here too, but maybe not quite as much as on aspen, but there's more on the maple. And so I guess that's what I was very.
I ask a follow-up.
Sure.
There's conifers at Sugarloaf as well, and
Yeah.
Rulania exists on conifers.
Yeah.
Is it just that the species doesn't grow on conifers? Why did you exclude?
Yeah, so they do grow on conifers. So Rulania is also present on, let's see, balsam fir, cedar, thank you, and then white pine. Yeah, but it's, so we had those species included in the sample, but the samples, or the sprays that were sampled were really low size in comparison. So I think the results remind me, Dr. R, the rotifers were still abundant, but not quite as high. But yeah, so they exist on conifers too, it's just not as big a population. Yeah.
Does the Rolania grow in other substrates like soil or rocks or?
It grows on rocks too. So Frulania as a genus will colonize trees and rocks. I excluded rocks from the sample, but I have collected Frulania from rocks, and I found some rotifers, but not quite as many as occur on plants. Yeah.
We have a question about the seasonality. The rotifers. So you found that you have higher density on the winter. Do you have any idea why it's like this? Are there maybe like less predation during the winter, or would that be linked to the behavior of dormancy of the rotifers that dedicate them to gather together?
Yeah, that's a good question. So, in my thinking about it, I attribute some of that to the moisture present in the winter. And just, I've collected rotifers in like the dead of February, on a sunny day where there's a little bit of snow kind of covering some Plania, and then find active rotifers when I bring it back to the lab. So I feel like the winter abundance is related in part to the amount of moisture that's in the air, these aspen stands, and also the stem flow and melting snow maybe falling down from the tree and falling leaves.
Right. Can I ask for a question? So basically, you sampled over years.
Wait, please say.
Sampled the 3 years. So with probably environmental conditions, do you have an effect of environmental conditions like temperature on the density of the winter?
I don't believe so. I'd have to phone a friend on that one. Do you have temperature data associated with the rotifer population?
Never been analyzed.
Okay. Yeah. It's hard because I put all of this together.
Could some of that just be the last round of reproduction in a good habitat? I mean, that's what I was getting at with the strict parthenogenesis and no males.
Sure, yeah.
That you would expect it to build up over time.
Sure, yeah, I mean, very well, so, yeah.
And then concentrate based on water liquid.
Yeah.
Winter.
I'm saving my questions for tomorrow, but,
Me too.
Oh, gosh, Oh, I was gonna bring it, but
you should,
oh.
there's somebody who makes it.
Describe it.
So my rotifer wrangling device. So this all has a purpose. So I collect rotifers with carbon, which is a microscopic tool that is made by a woman in Pennsylvania whose dad studied tardigrades. But it's essentially found her on Etsy, but some miracle. So the urban loop has a wire, and it's a really tiny little, like, basically a lug net for rotifers, but with no catcher. And so when you go in with this urban loop, you can use the water tension to kind of get a rotifer in the loop, and then it kind of balls up, and it's just in the urban loop, and then you can do stuff to it.
So you lasso.
Yeah, you wrangle. And then I just found a ton of tardigrades throughout this experiment. So Caitlin thought it would be funny to put me on a tardigrade, and then I just took it and then ran, so. Any questions? Oh. Wait. Okay. I don't know how to do that. How do I do that? Oh, here we go. Oh. Okay, no question. Oh, okay. Well, there's a class, so we have to end. Thank you all for coming.